Hello and welcome to the first of a series of videos on cell biology, today starting with the introduction to cells. And throughout the presentation I'll be talking about the cell theory, the unicellular eukaryotes, surface area and volume, cell sizes, magnification and scale, as well as multicellularity and stem cells. I hope you enjoy. So first of all, the cell theory is defined as, in biology, cell theory is the historic scientific theory now universally accepted that living organisms are made up of cells, that they are the basic structural unit of all organisms, and that cells come from pre-existing cells. And that's the fundamental idea of biology, and it's also closely linked to the development of the microscope in the 1600s, and the term cell was coined by scientist Robert Hooke as he inspected a tissue and saw that it was arranged on the microscope as cells of a monastery would be. So moving on, the main principles of the cell theory are first of all the composition, meaning that all living cells are composed of cell or cell compartments. Then we have the division, meaning that new cells are created by division of already pre-existing cells. Then we have genes. Cells contain inherited information used as instructions for functions. We have the unit, meaning that cells are the functioning unit of life because of the chemical reactions within. And of course, there are exceptions or caveats to that. For example, we have the striated muscle fiber that forms from a fusion of cells and has multiple nuclei. We also have giant algae that is one multinucleated cell and is maintained by only one cell wall. And then we also have some specific types of fungi that form strings called filamentous hyphae, where many nuclei are existing without any cell walls in between them. But moving on, we come to the life functions. First of all, we have homeostasis, meaning that there's a stable internal environment through chemical reaction, through continued series of chemical reactions. We also have nutrition, meaning that the organism, in order to be alive, requires food to turn energy to power for chemical reactions and to build cell compartments. We also have growth, meaning that it increases its size until it can divide. We have reproduction, that it divides to produce new cells, and in unicellular organisms, it produces genetically identical cells. In multicellular diploid cells, we have a continuous germline of haploid gametes. We'll also be talking about that when we come to genetics in a few videos. We also have the response, also sometimes called sensitivity, to environmental stimuli, and the receptors in the plasma membrane detect molecules in environment and send signals to the internal machinery of the cells. We also lastly have the metabolism, meaning that there is a continual series of chemical reactions sustained through nutrition, and sometimes it's a separate point, but here I've written it as a continuous point of metabolism. We also have excretion, which is the removal of waste product. And if you combine most of those letters together, you get Mr. Sheng, which is sometimes used as a form to remember the life functions of carbon life as we know it. Now, moving on to unicellular eukaryotes. First of all, we have the aspect of diversity, meaning they are able to perform all life functions with large amount of diversity in the way that they perform them. We also have single-celled organisms that comprise the majority of the diverse kingdom, and features, meaning they have generalized and specialized features. For example, most Protista that I'll be talking about can reproduce sexually, but most of them can as well reproduce asexually by binary fission. So we also have this aspect of diversity. And coming to Protista, Protista are eukaryotic organisms that are not plants or animals, are fungus, and they're most often found in water, and all the characteristics of unicellular eukaryotes can also be applied to Protista. And here I have two examples of unicellular eukaryotes. One, we have the paramecium, which is a heterotrophic eukaryote that has different organelles for the functions of life. So we have the cilia, which are hair-like structures with which it's able to move. We have contractile vacuoles that regulate water balance. We have food vacuoles for food storage, nutrition, metabolism, all that. We have the anal pore for excretion. We have two separate nuclei with both having different functions. It feeds on bacteria, and it also has an oral growth for ingestion. On the other side, we have the, I don't really know how to pronounce it, Cynedicin? I'm not quite sure, I'm sorry, which is autotrophic, and it synthesizes its own food. And heterotrophs and autotrophs are also going to come up when we'll be talking about ecology. But yeah, moving on, it has in its structure starch grains, a cell wall for structure formed of cellulose and pectin, chloroplasts, nucleus, paranoid, protective spines, and a vacuole. So as you can see, both differ in their organelles. Now, moving on to the third topic, surface area and volume. The key idea is that diffusion is less efficient in cells with a small surface area relative to volume than cells with a large surface area relative to volume, and small cells and small organisms generally have a large surface area in comparison to volume, whereas big organisms have big cells have a small surface area relative in comparison to volume. And diffusion is no longer effective when there is a physical limit. But wait, so we have the single-celled organisms that maintain their or meet their cell requirements by diffusion and active transport through the plasma membrane that regulates movement of substance in and out of the cell, and each square micrometer there's a limited amount of particular substance that can pass per second. And on the other hand, we have multicellular organisms, for example plants and animals, that require specialized systems to transport the materials to cells and tissue. For example, the acquirement of respiratory gases by diffusion through skin, or gas exchange through the lungs, or circulatory systems through the blood. And when diffusion is no longer effective in bigger organisms, because there is a smaller surface area and diffusion is no longer effective, as I already said, there's a physical limit on the maximum growth controlled by diffusion. And so large organisms become multicellular through a process called cell differentiation, which I'll come to in a second when we talk about multicellularity. But before we come to that, we have the cell sizes. So cells vary immensely from 2 micrometers to 100 micrometers generally, and prokaryotic cells are 10 times smaller than eukaryotic cells. And now an important thing when we talk about cells, an important instrument is the microscope. And the most common light microscopes can magnify a maximum of 2000 times, but an electron microscope can magnify a picture. What's that? Can magnify a picture 500,000 times its amount, because there's a better resolution because of the shorter wavelength of the electrons, and the resolving power also means the ability of a microscope to separate different structures visually. We also have two types of the electron microscope. We have the TEM a transmission electron microscope, and we have the SEM, the scanning electron microscope. And the difference between the microscopes and all of that and its history, I'll come to talk about more in detail in a different video. Here I also have the units of length in a table. So we have 1 meter equals 8,000 millimeters, 1 millimeter equals 1,000 micrometers, 1 micrometer equals 1,000 nanometers, and 1 nanometer equals 1,000 picometers. So you have an understanding and an overview over the different units. Magnification. Magnification means basically how much larger an object appears compared to its actual size, and it's the ratio of the size of the image to the size of the object. And here we have the two formulas used in scientific work. So for the magnification, you take the measured size of the object and divide it by the actual size of the object, whereas for the actual object size, if you want to calculate that, you use the size of the image divided by the magnification. And for example, if you have linear magnification, you first, if you have a worksheet, right, with a picture of a cell and next to it a scale, you first measure the object with your ruler, then you measure the scale line, and you can already calculate with the magnification the amount of magnification. But if you want to calculate the actual object size, you use the second formula. And with that we come to multicellularity, meaning that there are specialized cells that arise through the process of cell differentiation, regulated through the differentiation gene expression. And cell differentiation means basically the expression of some genes from the organism's genome in the cell, but not others. And through complex interactions of cells in multicellular organism results the emergence of new properties. And in multicellular organisms, cell differentiation has a specific hierarchy of biological order, and life is emergent of the property of chemical reactions driven by input of energy that produces work and results in entropy or disorders in a system. And the aforementioned hierarchy of biological order is seen here. So first you have, in multicellular organisms, a cell differentiation. Then we have the specialized cells with their, specific functions. They associate to form tissues. From then on, they organize into organs. And that's how the hierarchy of biological order comes and how multicellular organisms arise. And here I have a picture of cell differentiation all the way from the zygote to the different organs. And in the development, we have an unspecialized stem cell of a zygote that develops along the germ line, the mesoderm, the endoderm, the ectoderm, to form a specialized cell. Each cell has the same genetic material but different gene expressions to choose the cell type. It cannot change the cell type after the development. And if we look at, for example, from the embryonic stem cells, we go along the germ layers, and if you, for example, look at the endoderm, we have the possibility that the cell specializes into a pancreatic cell or a hepatocytes, and the endoderms, pancreatic exocrine cells and nerve cells, for example. So a lot of specificity and a lot of room for different possibilities, and there are over 230 cell types through cell differentiation, and that's only the types. Stem cells, interesting. The properties of stem cells are characterized as unspecialized and undifferentiated, so they do not go at first to the process of cell differentiation. They divide repeatedly into a large number of cells, which is why they're also called that they're able to have self-renewal. They also can differentiate into different types of cells due to their potency, and they have a large nucleus relative to the volume. And here in the middle you also see a picture of a stem cell. And there are different levels of potency. First of all, we have the multipotent stem cell that is also found in adults, for example, in the bone marrow. It can give rise to limited cell types related to the organ of origin, so bone marrow, and there are also specific stem cells found in the gut, and they can differentiate into specific cells that are also within the organs of the gut. Then we have the next higher potency, namely the pluripotent stem cells that can give rise to all cell types except the extra embryonic cells. And that's, for example, found in the embryonic stem. And lastly, we have the totipotent cell, the superhero among the stem cells, that can differentiate into all cells. And for example, that's the fact for the zygote and also plant meristematic cells. And they also play a large role, so stem cells play a large role in blood cell production, for example, with the help of multipotent stem cells that through differentiation and mitosis and controlled by a specific growth factor divide into one daughter of a stem cell and also a precursor cell that then becomes a cell necessary for blood cells, so a form of blood cells basically. Then we have the adult stem cells and the embryonic stem cells. And adult stem cells are undifferentiated cells in certain tissues of adults, children, and the umbilical cord blood. They're multipotent, so they can give rise to only limited cell types. Again, related to the tissue of origin. And therefore, because they're found in adults, there are less ethical issues with it, and generally experimentation with these kinds of stem cells are more accepted amongst international science. On the other hand, we have embryonic stem cells that are from the inner cell mass of the blastocyst, so you can find that along the membrane is a form of ball, I believe, and they are pluripotent, so they're not even totipotent, they're pluripotent, and they can become anything except the placenta cells. They also retain potency for multiple divisions, which is remarkable for cells, and they also have a therapeutic use in regenerative medicine and tissue replacement, for example, for Stargardt's disease or type 1 diabetes. And because they include the deliberate creation and destruction of embryos, it's in some countries considered unethical, for example, Ireland or Germany. We also have embryonic stem cells, on the other hand, that can come from cloned embryos using somatic cell nuclear transfer. So there's a patient match, and we don't have the issue of histocompatibility. And this histocompatibility is necessary for tissue transplant so that the body does not start to attack that body as an outer body, not a new part of the body. Similar to blood transfusion, you also need to have a matching type in order to not be rejected by the body, and same goes for tissue transplants. And through that embryonic stem cell cloning, this issue of histocompatibility can be avoided. There's also this therapeutic cloning with stem cells that is a solution for this histocompatibility. That produces then genetically matched stem cells that are totipotent. So that is something that is widely researched, although in some countries, in others not, because of the aforementioned ethical problems. And they have found, or they are researching many ways that they can turn it into therapeutic use. For example, for Stargardt's disease, which is an inherited juvenile macular degeneration, leading to a dysfunction of the retinal pigment epithelium, something in the eye that turns into a blind spot, I believe. And these cloned embryonic stem cells are cultured into the retinal pigment that is dysfunctional, And when they're injected into the eye, they develop and replace the patient's damage, so they can avoid the inherited juvenile macular degeneration of Stargardt's disease. So that is one example for possible treatments with stem cells, and there are many more interesting developments within that region. But yes, those are the basics you need to know for stem cells. And here's my bibliography. For these kinds of presentation, I'm mainly using the IB books that you can see down there. So the Walpole one, Biology for the IB Diploma, and the IB Biology Student Workbook. The other ones are extra information I needed for pictures and definitions and all that. So keep that in mind when you watch my following presentation if you want that. And I thank you for watching and have a nice day.